Okay, so first things first, we want to check whether what our data look like and where our data are stored. So I've downloaded the data, so you need to make sure that you also have the same data structure or data files downloaded from our class SFTP server. Okay, I've downloaded my data into this folder on my desktop, and I called it REP for repetition, because the study for which the data are collected are for a repetition experiment paradigm. If we open it up, you can see the example data. I have right now only downloaded 6 subjects worth of IMA data. All right, so this is from 001 to 003 and Y01 to Y03. These are young and older adult subjects. And in each folder, let me just focus on one younger adult first. You'll see that you have an AX2. This is what it means is it's axial T2 image. And then MP range, this is the T1 image. And then a Rep1 and Rep2 folders, these are where the functional EPI images are kept. If we expand this a little bit more, you can see that in the AX2 folder, there are, let me just go in there are 32 files, 32 IMA files. And what it means is that each file is one brain slice, one axial slice. So that means that there are 32 slices that make up this this T2 image. And if we were to go into the MP range or the T1 folder, you will see that there are 192 .ima files. These are the 192 sagittal slices of the IMA Dicom images for the T2 scan. Finally, if we go to the Rep1 folder, you'll see that there are 300 items here. Inside, actually, there are 2 sequences. If I can expand this, you'll see that there is a series that has this index here, which is rep.23. If you scroll down somewhere to the middle, you'll see that the 23 becomes a 24. What this is that actually the original EPI Dicom images are these rep23 sequences. And if I select all of them, you'll see that I have selected 150 .ima Dicom files. What this means is that there are 150 EPI scans that were actually collected for this rep1 sequence. And that means that we have 150 3D volumes. Remember that the EPI is a 4D image. It's every IMA file is made up of 2D slices of the whole brain. And so that you can imagine that if it is made up of one file has many 2D slices, that means that one file is effectively a 3D, has 3D information, 3-dimensional information in there. And then it goes on to the next file, which is itself also a 3D image, and another 3D image, and another 3D image, so that the whole sequence is a 4-dimensional data set. And this 23 here is the original 150 slices or 50 scans or measures, EPI measures that were acquired at the scanner. But what happens with Siemens scanners is that after it acquires the original image, Siemens scanners will automatically apply a motion correction operation onto the original image and then generate a new sequence. And then, in this case, it is sequence ID number 24, which also has 150 files. So the Siemens system will act on the repetition 23 sequence to apply motion correction on it automatically and generate the rep 24 sequence, series 24 sequence. For the purposes of this class, we are going to ignore the subsequently generated, Siemens-generated sequences. We will only use the original sequences. So for all rep1s rep1 data that you will see for all subjects, make sure that you always select the earlier number of that in that folder, the earlier sequence number. So in this case, we will only use rep.23. If we were to go to, that was rep1. If we go to rep2, we will only use rep.25, and you will see that there are also 150 of those up to here. 150 of the rep.25 series, and we will ignore rep.26. All right. Now we will do this for all subjects. So in Y01, this is the sequence number. Rep23 and rep.25 are the sequence IDs. These numbers are not always the same across subjects. This is because sometimes the subjects might have some error in the scans, and so they had to repeat some scans, or for whatever reason, they had to do some extra scans in between, so that maybe for a different subject, the rep2 or the second EPI run for that subject may not be sequence number 25. It may be sequence number 27 or 28 or 30. Depending on how many sequences were done prior to acquiring this second run or whatever sequence at that time. So what's important is not this actual number exactly, but what the relative position of it is in terms of the whole scanning protocol. So for example, we know that rep1 will always happen before rep2. So we will use whatever is the first sequence ID, in this case 23, for representing this subject's rep1, and this sequence ID .25 for this subject's rep2. Okay, for other subjects, we will have to go and take a look and see what is the number that was used. So let's see Y02 if that was the case. So Y02 also has the T2, T1 MP range, rep1 and rep2 EPI images. Let's take a look at Y02's rep1 and see if it uses the same sequence ID, and you can see immediately that's not the case. So for subject Y02, the first rep1, the first run, has the sequence ID rep.31. Okay, so that means that for subject Y02, there might have been a few other sequences that were collected prior to acquiring this rep1. And that's not important, as I said. What's important is you know that this is rep1 because it's been placed in this folder, and then you have sequences, the sequence number that is the original one is .31 because it's the earlier one. And there are 150 of these, and then Siemens automatically generates .32, sequence ID 32, and we don't want that. All right, so this is an example. Just to drive home the case now, let's look at rep2 for Y02, and you can see that here the ID, sequence ID is .33, which is what we want. The 150 .33 images that we want to use later. And we will not use .34. Okay, so this video clip is to orientate you to the Dicom image data set that you download. You will have many more subjects than I do. I think you have 17 or 19 young and older adults together, close to 37 or 40 subjects altogether. You need to download all of them. I will only be using these 6 subjects because in this class this is a demo that I am showing, so I won't be using the entire data set. And you will see that I have basically copied the data into this kind of data structure that I have. I have in my folder my project REP folder, I have set up a data folder, and then I have set up a source IMA folder under which I have a subjects folder and then the actual subjects data. You don't have to do this in this way. You can put the subjects data in whatever directory structure that you are comfortable with. This is just a standard practice that we do in our lab. It is closer to what we call the BIDS structure, and I will tell you more about that perhaps in class. That's not really a necessary thing, but this is something that you can try to use. And I will expand on this later on because here we are talking about the source IMA or Dicom images, and then later on we will also have the behavioral, so the source behavioral images, and I will talk about that in another video clip.